Antibodies, Chapter 12, Question 4. So an antibody is also known as an immunoglobulin. It is secreted from plasma membranes. A plasma cell makes antibodies that all recognize the same epitope. They are produced in response to bind to antigen that triggered B cells activation. It can activate complement cascades, neutralize antigens, and promotes phagocytosis of antigens. Antibodies neutralize antigens to prevent them from interacting with host cell. The structure. The antibody is a single monomeric structure. It consists of 4 polypeptide subunits. 2 are heavy chains and 2 are light chains that are held together by covalent bonds. If viewed as a two-dimensional structure, it has a Y shape. Tips of the Y-shaped molecule are antigen binding sites. This structure allows antibody molecules to carry out dual functions like antigen binding and biological activity mediation. The N-terminus of each heavy chain associates with one light chain to create 2 antigen binding domains. They are the arms of the Y-shape that are termed fragment antigen binding Fab domains. C-termin of the 2 heavy chains combine to form fragment crystallization FC domain. It represents the tail of the Y shape. It is important for the antibody's interaction with the effector cells like macrophages and activation of complement cascade. And these are the antigen binding sites, which are the arms, and these are the FC domains, which is the tail. So a Fab fragment binds to antigens. It is composed of 1 constant and 1 variable domain of each of the heavy and light chain. The domains shape the paratope, which is the antigen binding site, at the amino terminal end of the monomer. The FC region is the tail part of the antibody that interacts with cell surface receptors, which are FC receptors. This property allows antibodies to activate the immune system. There are 5 antibody classes. There's immunoglobulin G, which is the most abundant antibody in the human blood. It constitutes of 85% of total antibodies. It can be found in all bodily fluids, but it is especially abundant in blood, lymph, cerebrospinal fluid, and peritoneal fluid. It has a half-life of about 21 days in circulation before breaking down. It is one of the most versatile antibodies the human body can make. It is the only antibody that can cross the placenta to protect the developing fetus during pregnancy. It is a key activator of complement proteins, neutralizes antigens, and is a powerful opsonin. It has 1 Y-shaped molecule, so it is known as a monomer. The second one is immunoglobulin A. It is prevalent in mucus like mucous membrane of the gut, respiratory tract, and urogenital tract. It can also be found in most bodily secretions such as tears, saliva, sweat, and milk. It represents up to 15% of the human body total antibodies. It has a half-life of about 6 days before it breaks down. It can exist as a monomer or a dimer, meaning it has 2 Y-shaped molecules. The form of a dimer is resistant to harsh environmental conditions. It is an ideal antibody to protect nursing infants. It survives stomach acidity and can reach the intestines to confer protection. However, it is not good at complement cascades, but it has a good capability of neutralizing and opsonization. The third one is immunoglobulin M. It is mainly in blood. It is usually made early in infection upon a primary antigen exposure. Its presence indicates a recent exposure to a given antigen. It accounts for up to 10% of the total antibody population. has a half-life of about 10 days before it breaks down. It can exist as a monomer or a snowflake-shaped pentamer with 5 Y-shaped subunits. The large structure is central to agglutination and precipitation reactions that promote phagocytosis. It has also an important role in complement activation. It has some ability for neutralization, but it is not as effective as that as immunoglobulin A or immunoglobulin G. It is also not a strong opsonin. The fourth one is immunoglobulin E. It is present in very low concentration and mostly in lungs, skin, and mucous membranes. It is not central to complement activation or agglutination, and it's not a good opsonin. It is a monomer with a half-life of only 2 days before breaking down. It is central to fighting parasites and mediating allergic responses. Its role in allergies relates to its specific ability to encourage mast cells and basophils to release histamine, leukotrienes, and other allergy mediators. We can find an elevated IgE in patients with asthma, allergies, eczema, dermatitis, and certain parasitic infections like those caused by ringworms or tapeworms. The fifth antibody is immunoglobulin D. It is mainly found on the surface of B cells, and there's only small amounts that can be found in the circulation in the blood. It is a sparsely represented antibody with a half-life of 2 days before breaking down. It is a monomer-shaped Y molecule, and its precise function still remains unknown. And these are my sources.